All right, so let's go over this. So you had to figure out whether or not these two genes were linked or unlinked. And so the information that they first gave you is that the parents were one parent was big N, little h, and then the second parent was little n, big H, little h. And so no matter if the genes were linked or unlinked, all the kids are going to be heterozygous because this parent right here is always going to give a big N, and this parent will always give a little n, this parent little h, this parent big H. So you'd expect all the kids to be like this. And so what happens is the results are a little bit different if the genes are linked versus unlinked. And so let's compare this and see what would happen if they were linked or unlinked. So let's do linked first. So linked means that the genes are on the same chromosome pair. So I'll do this in blue to kind of keep everything together here. So if these two genes are linked for this parent here, that means that this parent has a chromosome pair, let's say pair number 1, and they have big N's on each of them and also little H's. So they're linked on the same chromosome. This parent would have little n's and big H's here. So then we'd get the same kid because this parent would pass on either this chromosome or this chromosome, either one. They're going to pass on a big N and a little H, chromosome number 1, and then this parent's going to pass on a chromosome number 1 as well and be a little n and a big H. And so therefore the kid is big N, big H, little h, and that's what we have here. So then what comes into play next is that you have to do a test cross here. So we're going to do multiply, cross it with a little n, little h kid. So if these genes are linked, you're very powerful.
That is honestly a fire animal, all right? Let's be honest.
This one?
Yeah.
It keeps going every time you touch it.
Yeah, I probably should put a
Every time you touch it, it's turning.
Yes, all right.
I thought it would help plugging it in.
Let's try. Let's try. Let's plug it in. Let's do a different plug.
Every situation when I try to help, it just totally.
Let's try this one.
Very powerful.
What can I say? I'm magic.
Alrighty, so going back to these two chromosomes would be little n, little h, little h. Let me make that. So that's their chromosome number 1. So now if they're linked, drawing this out helps to think about how they're linked. And so we can make a Punnett square, but when we look at this parent there's only 2 combinations that they can pass on. The chromosome here with the big N and the little h, or the chromosome over here with the little n and the big H. So therefore we have big N, little h, or little n, big H. And then this parent is always going to pass on a chromosome number 1, but no matter which one they pass on, it's going to have a little n and a little h. So therefore we have a 2-by-1 Punnett square, where this is the result. So we'd have dominant and recessive, and then recessive and then dominant. And you'd expect a 1-to-1 ratio. And so when you look at the data that you had, most of the kids from this, it says the one that says 93, the 93, when it says nice and very ugly, that nice is a dominant, and then there are 2 recessives, so that's like this one. And the other one that's 97, that's in high quantity, is mean, which is recessive, little n, and handsome, which is a big H, something, which is this one here. So we get those 2 phenotypes, but we also get some that are in smaller quantities. We have the 6, nice and handsome, which is all dominant, and we get 4, mean and very ugly, which are all recessive. So we get some extra here. So these ones are the ones that you'd expect here, but we get extra. So then if they're unlinked, let's look at if they're unlinked. I'll do this in a different color. So if the parents here, if this parent here, if the genes are unlinked, that means that they're on chromosome pair number 1, let's say, they're big N, big N. And unlinked means that the H's are on a different chromosome pair. So therefore, the H's are on, let's say, chromosome pair number 2. And then this parent is little n for chromosome number 1, and big H for chromosome pair number 2. So now when these guys have kids, and they're also going to have all heterozygous kids, because this parent's going to pass on 1 chromosome number 1, and this parent's going to pass on 1 chromosome number 1. This one has to pass on a big N. So this parent over here is going to have a chromosome number 1 with a big N from this parent, little n from this parent, and then they're going to get a chromosome number 2 from each parent, and then they're going to get a little h from this parent, and a big H from this parent. And so they are all heterozygous, big N, big H, little h. So now when they cross it with the homozygous recessive, the little n's are on chromosome pair number 1, and the little h's on chromosome pair number 2. And so now, this is where we're going to look at here, the data. And so now what happens is if they're unlinked, we have more combinations that this parent can pass on, because they're going to pass on 1 chromosome number 1, 1 chromosome number 2. So they can pass on a big N and a big H. Oops, I skipped one. Or a big N and a little h. Or a little n and a little h. Or a little n and a big H. So we have 4 combinations for this parent, which we didn't have before when they were linked. And so now this parent, though, since both chromosome number 1s are a little n, they're always going to pass on a little n. And since both chromosome number 2s are a little h, they're both going to pass on a little h. So that's the only combination that parent can pass on to their kids. So we have a 4x1 Punnett square here. You could do this 4 different times and have 16, but just each row would be the exact same. So you end up with a 1 to 1 to 1 to 1 ratio, where you get all 4 combinations that we see here in the data, but it's not in a 1 to 1 to 1 to 1 combination. So this trait most likely is not unlinked because you have such small numbers in these two. So what gives us the trait, these genes are linked? What helps us to understand that they are linked? Well, we did the test cross and we saw what we'd expect, but these results aren't exactly what we expect. So what are we looking for to explain that they're linked? Like how could you tell that they're linked? Most people, as I walked around, said that they're linked. But my question is, if you had to write and justify why they're linked, what would you say?
Is it like if you have 2 numbers that are really high and then 2 numbers that are really low, you can tell just because that's so drastically different?
Yeah, that's the first thing is that the numbers that you have really close to, you have the highest numbers. The key here is that not only looking at 2 numbers that are really high and 2 numbers that are really low, that is an indication, but the 2 numbers that are really high should be like the 2 that you would expect with linked, all right? So therefore, that's what you're looking for here. Notice that these 2 are like what you would expect, and therefore these low numbers are only due to a situation where the genes had to be mixed around. So these guys here are the ones that are due to crossing over. So really the justification is the 2 high numbers, those kids are like the expected that you would get in the linked cross, okay? And so then it asks you, so we didn't get a 1 to 1 to 1 to 1 ratio. If we would have gotten a number like 93, 97, 90, and 99, what would you have told me? They're unlinked, right? All right, we did not get that. And so then to figure out your percent of crossing over, another way to say that is recombination frequency. So what you do is you take the number of individuals that can only happen due to crossing over, which are these guys here. So you have 10 are what we call recombinants. And we divide by the total number of offspring. So we had 10 of them that are due to crossing over out of our total, which is 200. So 200 total offspring. And then we take that to get a percent. We multiply by 100. And if we round up, it's about 5%. Okay? And that's your recombination frequency. So what does that tell us? That between those two genes, since they're linked on the same chromosome, between the N and the H gene, that they switch and crossing over occurs about 5% of the time. So 95% of the time it doesn't occur. And that's why we see so many individuals that are what we would expect with no crossing over. Okay? How'd you guys do on that? Okay, good? Alrighty, perfect. Alright, so today we're going to wrap up chapter 15. You able to get that out? You're gonna get it out. So, yeah,
I was gonna say, I feel like the jeans, it's gonna hit right on the little, like, you know, the belt with the right on your tailbone. That's why. Especially in jeans. No, more baggy. I will not be calling her Mark. No, Mark. The G bar. Yeah, I'll go with Peggy. Yeah, I won't respond to anything.
All right, you guys. I think this is the picture we left off at. Yeah. And so we were talking about how you can get abnormal numbers of chromosomes, and it has to do with mistakes during meiosis. Those mistakes are called nondisjunction, where the chromosomes don't separate properly. And I think in this class we only talked about the left side yesterday, where the homologous chromosomes are supposed to go into opposite cells in meiosis 1, and these two both go to this cell. Right? We talked about that? All right. And so you end up with egg cells that have too many chromosomes or too little, have an extra or missing a chromosome. And then over here shows you the second way that this can happen. Meiosis 1 can happen properly, so the homologous chromosomes separate, but meiosis 2 doesn't happen properly. What's supposed to happen in meiosis 2, Roman? That's right. And so therefore, in this case here, notice that there's not a spindle attached to this side, this one from the side. And so both the sister chromatids end up in one cell, whereas the second cell here ends up with none. And so these two are like these two here that we talked about yesterday. And then in this cell, meiosis 2 can happen properly, and so there's nothing wrong with these two gametes. All right? And so you can end up with missing or extra chromosomes. Yesterday I draw like a sperm cell and show if they unite to zygote. Like, did I? Okay. All right. So aneuploidy results from the fertilization of gametes in which nondisjunction has occurred. Offspring with this condition, aneuploidy, have an abnormal number of a particular chromosome. Particular. So, like for us, we can't have 3 of every kind of chromosome, but there are certain chromosomes that we can survive with 3 of, but have 2 of every other kind. And it depends upon which one we have 3 of, what symptoms we have. So, a trisomic zygote, tri meaning 3, has 3 copies of a particular chromosome. A monosomic zygote has 1 copy of a particular chromosome. And polyploidy is a condition in which an organism has more than 2 complete sets of chromosomes. So, an example of a trisomic zygote, is trisomy 21. This means that the individual has 3 copies of chromosome 21. So somewhere either in the mom or the dad, most likely it's the mom we're going to see when we talk about trisomy 21, that it's most likely due to non-disjunction in the mother, and therefore the mom and her egg cell give 2 chromosome 21s, and dad gives 1, and the baby then has 3 chromosome 21s. This is called Down syndrome as well. Oops. An example of a monosomic, mono meaning 1, so it has 1 copy of a particular chromosome is, do an example here. They call it monosomy X, or they, another way to say, they talk about this is called Turner syndrome. So monosomy X, or Turner syndrome, what do you think? What do you think is going on with this person's chromosomes? Lucas? Okay, so in what chromosome pair is this affecting, do you think? The sex chromosomes, that 23rd pair. So monosomy X is for your sex chromosomes, your 23rd pair. People with this, they call it X0, meaning that they don't have a second sex chromosome. So females are supposed to have XX and males are supposed to have XY. People with monosomy X just have 1 X chromosome. They don't have a second, either X or a Y, so they don't have a pair there, hence the monosomy. All right, they're missing, they only have 1 copy of a particular chromosome. Would this person be a male or a female? Female. Maleness is determined by the presence or absence of a Y chromosome. All right. All right. So these guys are female, and so we'll talk more about these a little bit later. Polyploidy. This occurs in plants. This does not occur in us. Polyploidy. Yeah, for some reason for us, it says polyploidy, a condition in which an organism has more than 2 complete sets of chromosomes. We have 2 complete sets of chromosomes. 1 set that we got from mom, 1 set that we got from dad. That's why we have pairs of chromosomes. So we cannot survive. If we got an extra chromosome, let's say there was extreme non-disjunction in, let's say, mom's egg cell. So mom gave pairs of chromosomes. So she gave 46 chromosomes, 2 of every kind, and dad made his sperm cell fine. He gave 1 of every kind of chromosome. So then what happens is that you would have a baby with 3 of every kind of chromosome, or 3 sets of chromosomes, and in humans, the zygote would die. But for plants, they don't. And so plants, we'll look at a little bit later in the course that you can have plants ones that are diploid and ones that that are different. Some are triploid, and they can mate and actually create new species of plants and so on with different numbers of chromosomes and so on. And plants are able to survive. So when it says more than 2 complete sets of chromosomes, so an example would be the plant is 3N or 4N, meaning they have 3 or 4 sets of chromosomes instead of 2 like us. Okay, so this is abnormal numbers of chromosomes. The second thing that we were going to look at is how we get genetic disorders is by having the right number of chromosomes, but something being structurally wrong with your chromosomes. So there are 4 things. There's deletion, which removes a chromosomal segment. Duplication, that repeats a segment. Inversion reverses a segment within a chromosome. And translocation moves a segment from 1 chromosome to another chromosome. And so if we look at the next picture here, the next picture shows you these 4 different alterations in the structure of the chromosome. So what you're looking at here, so here's the names of them: deletion, duplication, inversion, and translocation. On the left, you're looking at the chromosome, original structure of the chromosome. On the right, what the chromosome looks like after one of these things have happened. And they highlighted in the darker purple the area of the chromosome that's affected. And to make it so that we can understand it a little bit better, what they did is, remember, chromosomes are made out of a piece of DNA, and what they did is just chop the chromosome or divide it into sections, so section A, B, C, or D, so that you can see what happens from original to after the deletion or whatever happened. So in this particular case with the deletion, you can see that segment D is totally deleted. So therefore, remember that DNA is the order of the bases, A's, T's, C's, and G's, and you have a whole bunch of genes. So in this section of DNA, in section D, there might be 10 genes in there. And so therefore, if you have that section deleted, you have 10 genes completely deleted out of your DNA. Okay, so that's your deletion. A duplication. So you can see here B and C gets duplicated. So now you have this piece of DNA, but you just have these genes, whatever genes are in this DNA, you have them twice. And so you have a little bit of a longer chromosome, and so you have 2 copies of the same genes on one piece of DNA. So if this is this chromosome here, this is replicated and this is duplicated, and then this is your second copy. But remember, that's one chromosome. And then if you have another chromosome and you have is not duplicated and you have this, then you have 3 copies of those genes on that segment of DNA. And that can be a big deal. Like, for instance, like Trisomy 21 with Down syndrome, they have 3 chromosome 21s, therefore have all 3 genes for all of the DNA in chromosome number 21. And you can see that, we'll talk a little bit later, that there's characteristic symptoms of people with Down syndrome. And so having 3 or extra DNA usually creates problems, usually in certain, and depending upon what genes that are extra that you have, create a different set of problems. An inversion, notice here, the section B, C, and D, it's like they cut the DNA out of this and flipped it around. So now here, so the B is on this side, so it's backwards, so it's DCB, DCB here. So you have your DNA, you still have those sections of DNA. Why is that a problem then, do you think? Why is it a big deal that it's flipped?
I don't know what those genes control. Like if they control something important, like a certain protein that has to be made, the protein will get made right, and obviously protein.
Absolutely. Now, why wouldn't the protein be made right? It's not coded right. Remember, it's a particular order of A's, T's, C's, and G's in your DNA that code for what amino acid is going to hook to what amino acid, and to make then the R groups interact and get the 3-dimensional shape of your protein. If you flipped this around, then you've changed the order of the DNA. So let's say it's read from left to right, and so the order of the A's, T's, C's, and G's make the right amino acid chain. But now we've flipped it, and now we've changed the order of A's, T's, C's, and G's, and we're going to make it wrong. And so kind of like you can read a sentence from left to right, and it makes sense, but if we took that sentence and flipped it around so you read it backwards and tried reading it left to right, it wouldn't make any sense. All right, and so the same type of thing goes with DNA. So inversions can be a big deal. And then lastly, we have translocation. This is a little bit different. Notice the original, you're looking at 2 different chromosomes. These are non-homologous chromosomes. So this could be like a chromosome number 9 and a chromosome number 22. And so they're not homologous, they're different shapes, sizes, and then they tried to show this by even segmenting the DNA with different letters there. So they're not the same, they're not a pair of chromosomes. And so what translocation, reciprocal translocation means that they flip pieces of DNA. This is reciprocal, meaning that this one this piece is going to go over to the end of this, and this AB is going to replace where the MNO was. So you can see that's reciprocal. Also, you can have non-reciprocal translocations where a piece of DNA can actually just translocate and add to the end there, and this one doesn't do the same. All right, so that's why that's a reciprocal one. And this can actually also affect, because if you look at this, if this is your original piece of DNA right here, and the rest of the DNA is supposed to be right here. All right, so this is the chain that's supposed to be, it was right here. So let's say that you have a gene that started right here, so this is the start point of the gene in the DNA, and this is the end point. Guess what, you just cut a gene in half, all right? And so therefore the rest of this gene is not on the right chromosome, you don't have an attacked gene, and therefore you can get, not make the right protein, and therefore you'll be deficient in that and have some repercussions from that. Okay, so that's the changing in chromosomal structure. So now we're going to take a look at, I'm going to give you examples of different actual genetic disorders that illustrate these. Yes.
This is caused by like environmental factors, right?
It can be, yes. Yeah. All right, so some actual human disorders. So we'll look at the abnormal numbers of chromosomes due to nondisjunction first. So alterations of chromosome number and structure are associated with some serious disorders. Some types of aneuploidy, that means the abnormal number of chromosomes, appear to upset the genetic balance less than others, resulting in individuals that are able to survive, they're able to be born and actually live. Some you get abnormal numbers of chromosomes and the baby can die while still in the mother. These surviving individuals have a set of symptoms, or what we call a syndrome, characteristic of whatever aneuploidy they have, or whichever one they have extra or missing chromosomes. So the first one that we've kind of talked about is Down syndrome. Results from 3 copies of chromosome 21. They've changed our book says 1 in every 700 kids in the United States. It's now 1 in 830. It's going down. It's good. Kids in the United States. They found a correlation, a correlation between the frequency of Down syndrome and the age of the mother. And so it seems to be more correlated with mom getting 2 chromosome number 21s and therefore having the mistake in meiosis and nondisjunction than dad in having children with Down syndrome. And so you can see here that under 30 is about 0.04%, and age 40 goes up to 0.92%. And so, so the likelihood or the chances of having a child with Down syndrome increases with the age of the mother. And they noticed that at age 35, which is kind of in between there, is kind of when from 35 moving forward, it gets increasingly higher percentage at that magic age of 35 for the mother. All right, and so so mom most of the time for Down syndrome, mom gave 2 21s, dad gave 1 21, and then the baby has 3 chromosome 21s. All right, so then there's characteristics. So I'll show a picture in just a second. There's some characteristic facial features with their eyes and so on. Usually shorter. They usually have some types of heart defects that are common. That most people with Down syndrome probably have had some kind of heart surgery in their life before to correct that. They have developmental delays, so it's harder to learn to do things and different milestones in childhood and so on. And can be sterile. It says may be sterile, not always. Sterile means unable to have children. All right, absolutely. Okay, so here's a person with Down syndrome. She, this is her karyotype. So this is a real karyotype of her chromosomes. You can see pairs 1 through 23. She has 2 X's to make her female, but notice here her chromosome 21, she has 3 of them. And so remember, so some people, if it's thought, sometimes when mothers are age 35 or above, doctors will leave it up to the mother to do certain testing to see if their baby has a chromosomal disorder. Remember last chapter we talked about it's the test where they stick the needle in and suck out amniocentesis. And so they suck out the amniotic fluid, which has fetal cells, the baby cells in it, and then you can get into the cells, get the chromosomes, and do a karyotype like this so they can tell whether or not the baby has the right number of chromosomes, and if not, which one do they have extra missing of, and therefore know what disorder before the baby's born. Sometimes that's helpful because you can have things in preparation for special care that the baby may need and so on with a specific syndrome or disorder. All right. This can also happen. So trisomy 21 is with the autosomes, but it also can happen with sex chromosomes. So nondisjunction of sex chromosomes produces a variety of conditions as well. So I'm going to introduce two to you. There's Klinefelter syndrome. This is the male having an extra sex chromosome. The male has XXY. So he got either mom gave 2 X's and dad gave a Y and he's XXY, or mom gave 1 X and dad gave an X and a Y to get XXY. And you can see the characteristics: small testes, sterile, they cannot have kids, some breast enlargement, affects their intelligence as well. So this is a type of trisomy because for that 23rd pair, you don't have a pair, you have 3, all right? And so that's an example of a trisomy. Turner syndrome, which we wrote earlier, is monosomy X, so they have this, just the X0 female, they only have the 1 X. They're also sterile, and this is the only known viable monosomy in humans, meaning that viable means living, able to survive, all right? So they're the only ones that are able to survive into adulthood and so on. So notice here, they're sterile, but they have normal intelligence, unlike the Klinefelter syndrome. And they only have 1 X chromosome, so this last part I put that they can be given estrogen replacement therapy. Because only having 1 X chromosome, they have lower amounts of the hormone estrogen. So males and females both have estrogen and testosterone. Males have a higher level of testosterone than estrogen, and females have a higher level of estrogen than testosterone. And it's these levels that help to determine secondary sex characteristics. So for instance, the higher amount of testosterone in males give males things like facial hair and a deeper voice, all right? And so those are secondary sex characteristics. And then the higher level of estrogen in females, breast enlargement and things like that are secondary sex characteristics. And so with the 1X for Turner syndrome, she may have lower levels of estrogen but can be given estrogen so that she can develop secondary sex characteristics as if she had 2 X's. And which is why up here with Klinefelter syndrome, the male with 2 X's can have some breast enlargement, which is characteristic of a female secondary sex characteristic by having that extra X. All right, and so that's that. Some examples of structural problems with the chromosome: the cry du chat, cry of the cat, results from a deletion on chromosome number 5 in humans. It's a horrible disorder because the baby usually dies in infancy or very early on in childhood. It affects the brain, the development of the brain and intelligence. It's called that because of the characteristic cry it has when the baby cries. It sounds like a cat cry, all right? Apparently, I have not heard a child, I'm not a child like this, but apparently that's what a huge thing that is characteristic of the baby. So that's a deletion, so that's an example of a chromosomal disorder with a deletion. And then translocation, some cancers like leukemia are due to a translocation. And so translocation, this is actually a reciprocal translocation. And so this is where leukemia is the white blood cells divide uncontrollably. Remember, cancer is uncontrolled cell division. And so this happens in a white blood cell that is going through mitosis, and then once this reciprocal translocation occurs, then any descendants of that cell will also have this translocation. So what happens? Here's chromosome number 9 and chromosome 22, non-homologous, and it's reciprocal translocation, so the end of the 22 replaces the end of 9, and the end of 9 goes to 22, so that's the reciprocal. So this chromosome 9 now is much longer than it was, and now this little chromosome 22 now has the end of chromosome 9. This is called the Philadelphia chromosome, but what they found is that the addition of that DNA onto the end of that and the deletion of this activates a gene that allows the cell to continue to go through the cell cycle and is not stopped, and therefore divides uncontrollably, and therefore hence what cancer is. All right, so so it activates a gene that does that. So that's an example of a translocation. So these things here are not due to anything that are inherited from parents. It happens usually within the individual. And the last thing I want to talk about with this chapter with genetics is some more unusual things. So everything that we've talked about so far has been DNA in the nucleus, and we're going to look at another exception that affects DNA in the nucleus, but then we're going to look at genes that are located outside of the nucleus. Where are genes found outside the nucleus? Where in the world do they? Yeah, the mitochondria. And it's not NUS. Chloroplasts. All right, so we'll look at these. So let's look at the exception here that's unique with still DNA in the nucleus. So whoops. So the situation called genomic imprinting. Few mammals and a few traits are affected by this. The phenotype depends on which parent passed along the alleles for the traits. And so therefore, you get this variation of phenotype depending upon which parent gave it, and that's called genomic imprinting. It involves the silencing of certain genes that are stamped with an imprint during gamete formation. So what does this mean? Everything that we talked about so far, like if you get a big R from one parent, a little r from another, if you're big R, little r, you're big R, little r, and you show the dominant gene, it doesn't matter if you get the big R from dad or the big R from mom, you're big R, little r. With these, there's a few genes that it seems to make a difference if you get it from mom or dad. And so this is this imprinting. And so what happens is that there are a few genes that one of the parents' genes is silenced, which means that it doesn't matter what's on that parent's chromosome that you got. You don't show, isn't expressed, or is not shown up. So when it says genomic imprinting involves the silencing, so this means that what that means is inactivated. So the gene, it's an inactivating certain genes. Some people were asking me earlier what silencing. And then it says that are stamped with an imprint. What that means is, what they do is they add methyl, let's write that out, methyl groups. A methyl group is CH3. So they add methyl groups to the chromosome. I'll just put to the C, which inactivates it. So they make it so it doesn't work, and you only do this with one of the two parents' chromosomes. So let me show you an example with this. This picture is of a mouse. And so let's look at what we're looking at here. So here's your mouse. So letter A here, it says a wild type mouse. So this is normal, a normal mouse. And this mouse has two chromosomes, a pair of chromosomes, paternal and maternal, one they got from dad, one that they got from mom. they have a gene here. It says normal IGF2 allele and normal IGF2 allele. So they're homozygous for the normal IGF2. What does this do? This is a gene that produces a growth factor. What do growth factors do? They promote growth, okay? So this mouse has two genes that are normal genes that produce this growth factor. So lo and behold, this mouse grows properly, and so it's the normal size. All right, so then when we look at B here, we're going to look at two examples with B. So I'm going to kind of look at this as, this is what example 1, and this is example 2. What these two mice in these two examples have in common and how they differ from the first mouse is that these mice are heterozygous. So look at this mouse. There are two chromosomes. They have a normal IGF2 gene and a mutant one. So this would be like big R, little r, okay, in this. And so they have a normal and a mutant. And then this one also has a normal gene and a mutant gene. All right, so they're both heterozygous. So everything that we've learned so far is that if you're heterozygous, you would look the same. But look at what happens with these two mice. This one is a normal size. This one is a dwarf, even though their genetic makeup is the same. What's different is who they got the normal gene from. So according to this, who do you want to get the normal gene from, mom or dad? Should be dad, right? Because look at here. This mouse has a normal gene from dad and a mutant gene from mom, but appears normal. This one gets the normal gene from mom and the mutant gene from dad, but doesn't appear normal, doesn't grow properly. So mom's chromosome here, even though she has the normal gene, it's not working properly. And so what that has to do with back to that genomic imprinting. What happens with this gene in mice is that the mouse does imprinting or adds methyl groups to the maternal chromosome so that nothing that's on the maternal chromosome actually shows up in the mouse. And so the mouse has the phenotype of whatever gene is on dad's chromosome. So it doesn't matter what's on mom's chromosome. So this is what it means by not expressed here. Mom's gene is not expressed, which means that this has been silenced or inactivated by adding those methyl groups. By adding methyl groups. And that is what the process is called, genomic imprinting. And so for this particular trait in mice, we only see what's on dad's chromosome, all right? And so, so that is, so that's a little bit different, okay, than in a lot of the other traits we've looked at. And then the last thing is we have these genes in other organelles. They're called extranuclear genes. So extranuclear genes are found in organelles in the cytoplasm, like the mitochondria, the chloroplast. There are certain traits that are controlled by these genes, but they depend upon the maternal parent because the zygote cytoplasm comes from the egg cell. The first evidence was in plants here of yellow and white splotchy plants. But let me talk about this second bullet here real quick. Why it matters when it says by the maternal parent. All right, let's look at humans here. So here's an egg cell. Mom has 23 chromosomes that she's going to give to her kid in her egg cell. Then you've got a small little sperm cell that also has 23 chromosomes. And the sperm comes to fertilize the egg. Basically, all the sperm does is give the 23 chromosomes. So the sperm gives 23 additional chromosomes, so the baby's going to have 46 chromosomes. And that's the role of the sperm. Sperm is now done with its job. So now this is not an egg, this is now a zygote now because it's fertilized. But where this baby gets all of its organelles, all of its mitochondria, all of its Golgi apparatus, and so on, is from the mom, because it was all in the egg cell. Because dad, with the sperm cell, doesn't give any organelles to the baby. So all the mitochondria and stuff stays within the sperm. So any mitochondria, and therefore genes in the mitochondria that this baby has, is due to the genes that mom had. All right, because dad does not give any mitochondria to the baby. And so now this baby will then divide into and be 2 cells big, and those mitochondria will be distributed to those 2 cells. Mitochondria do have the ability to self-replicate, so they can make more mitochondria, but those mitochondria originated with the mother. And so therefore, any genes in the mitochondria, you can trace from parents back to the mother, and from that mother to her mother, and that mother to her mother, and so on and so forth. All right? And so that's where I'm going to end here. What I'd like you to do is this sheet here, it's just a little 1-page article. I would like you to read that for Monday. Right? You guys really want to finish the last one?
That was spoilers.
Those people who really want to, I'm going to come back and talk about this thing on Monday, but those people who like to have
They're just dying to
finished,
know.
I know. Unfortunately, it's not the first to know, I don't think. It's just the first. Done.